Hi guys, in this video we are going to discuss the TS of dicot and monocot leaf. So let's start with it. First thing, you must know some examples of dicots. The examples of dicots are rose, mustard. Okay, so these are some examples. Then here you can see or imagine a leaf of rose. Okay, here you can see the midrib, veins and veinlets. Okay, now this leaf has two upper and lower surfaces which are different. So this type of leaf is known as dorsiventral leaf. Okay, now let us see the TS of this leaf, transverse section of this leaf. So if you observe it under microscope, you can observe this. Okay, now in this case, let's start with the first layer. The first layer or the upper layer of leaf, as you might have seen, is waxy layer. Okay, which is known as cuticle. Now what does this cuticle do? It prevents the loss of water by transpiration. Then next after this is upper epidermis, which is also known as adaxial epidermis. Now adaxial epidermis does not have stomata. Okay, then next after this is the mesophyll. Okay, the mesophyll is divided into two types of cells, that is palisade mesophyll, which are elongated cells without intercellular spaces, and the another type of mesophyll is spongy parenchyma, which consists of air cavities. These are large cells, okay, with air cavities. Now what is the function of mesophyll? As you can see, they are green in color. They consist of chloroplasts, so thus they carry out the process of photosynthesis, and that's the reason we say leaf is the site of what? Photosynthesis. Okay, now here you can see when the photosynthesis occurs, okay, they'll produce some food, okay, and at that time all those nutrients which are produced, they'll pass on through this vascular bundle to all the parts of the plant. Now this vascular bundle, that is red ones is xylem, blue ones are phloem. Okay, xylem carries water, phloem carries food. Then next, these are surrounded by the bundle sheath. These are large protecting cells. Now this vascular bundle is present along these veins. Okay, so these veins, veinlets, okay, in this at these places these vascular bundles are present, because veins conduct what all the nutrients and water. Now as you can see, the midrib is thick. Okay, so it has larger vascular bundle. The veins are a bit thinner than midrib, so they have a bit smaller vascular bundle. So the sizes of vascular bundles are different, depends on the size of the veins. Okay, so this is all about vascular bundle. Then last part is the lower epidermis. See, we have come from here to here. All the points are in the sequence. Then here you can see the lower epidermis. Here you can see the presence of stomata. Okay, now can you see substomatal cavity here, this part? Now when the water vapors are lost from this mesophyll, they will gather here and they will be lost. They will move from this substomatal cavity through stomata and they will be lost, and thus the exchange of gases can also occur. So that's it about dicot leaf. Let's move to monocot leaf. Now monocot leaf, first starting with the examples. What are the examples of monocot? Sugarcane, wheat, rice. These all are some grasses. Okay, so as you might have seen the leaves of them, you might have observed they show parallel venation. All the veins are present in the parallel manner. Now, and they all are equal in size. Okay, so let's see here. When you take the transverse section and observe it under microscope, you can see this. Okay, so let's see the structure of TS of monocot leaf. Starting from up again, the cuticle. Then you have upper epidermis with stomata. So let's make a star because this is different point than this. Then next you have the mesophyll. Mesophyll is different again. Can you see the visible difference? Here the palisade mesophyll is present. Here the mesophyll is not differentiated into palisade and spongy. There is only one type of mesophyll, that is spongy here. Again, these are green cells, they carry out photosynthesis. Then you have vascular bundles. Now vascular bundles, okay, they are all of equal in size. That's the reason you see here the parallel venation. Okay, so here all the vascular bundles are equal in size. Now next thing, the lower epidermis. Lower epidermis also shows presence of stomata. So here in both the epidermis there is stomata is present. Here also you have substomatal cavities from where the exchange occurs, exchange of gases occur. Okay, so now let's see one unique point about monocot leaf. Now this adactyl cells, that is cells of upper epidermis, okay, along the veins, whenever this adactyl epidermal cells come, these cells convert into what? See, this is a vein with vascular bundle. The cells along this vein, the adactyl cells along this vein become large empty cells. These are known as bulliform cells. Now these bulliform cells, they absorb water, okay, and the leaves are exposed, okay, now exposed. And when this water is lost, when the cells become flaccid, see, when they become turgid, the leaves open up and they are exposed, but when the cells become flaccid, the leaves curl up, they become curly, and thus the water loss is prevented. Okay, so that's it about dicot and monocot leaf. Okay, I hope you guys have understood this. For more such videos, do like, share and subscribe to my channel. Thank you.